While the pigmentation of skin, hair, and iris color can be different, they share many of the same genetic mechanisms, and so there are often aspects of these different forms of pigmentation which are inherited together. So, for example, there are a number of genes which will determine whether an immature melanosome becomes a pheomelanosome or a eumelanosome. In the most common and in what was the ancestral human condition, the hormone MSH, and to a lesser degree the hormone ACTH, interact with a receptor known as the melanocortin receptor 1 or MCR1. This causes the majority of the melanosomes to become eumelanosomes, producing the brown pigment. In some individuals, there is a mutation in the gene for the MSH hormone. This abnormally shaped hormone can no longer bind the receptor and can no longer stimulate the production of the eumelanosomes. These individuals would have lighter skin and lighter hair, often a red color, given that the pheomelanosomes should produce a red pigment. More commonly, however, individuals fail to make the receptor MCR1. Without this receptor, cells are not stimulated by the hormone MSH or ACTH to make the eumelanosomes and their brown pigment. These individuals would then have lighter skin and hair, often a red color, and a difficulty in tanning. To a large extent, Asian and European populations evolved lighter skin independently through different genetic mechanisms. One of the main genetic changes in Asians was changes to the MCR1 receptor, which resulted in lighter skin. There are also other signaling proteins involved, including some such as agouti signaling proteins, which are capable of blocking the MCR1 receptor so that MSH or ACTH cannot bind. And so there are inhibitory proteins which would cause less pigmentation in addition to those which would increase pigmentation. Hormones and their receptors determine which type of melanosome develops, as does a protein known as OCA2, which determines the pH of these melanosomes and affects the pigment which is produced. Other enzymes are also required, such as TRP enzymes and others for eumelanosomes. Those are homologs of tyrosinase. Additional cysteine is needed for pheomelanosomes.